Over to you, Sika. Maybe you have the answers.
Right. Thanks so much, David. Yeah, from my side, first, a few words about our society, actually. The GA is the Society for Medicinal Plants and Natural Products Research. And yeah, in general, what we want is to promote natural products in their research, but also their development. So we believe that this can be achieved by an evidence-based approach of natural medicinal use. Because I wanted just briefly to remind you that we are a society for researchers, both in academia but also in industry, and for them we provide a worldwide support network for the members. But also we are a growing society. And for this reason, in the recent years, we have established a community within the GA, which focuses specifically on the African continent. And that's the African Research Network. So we organize, for example, symposia, summer schools, and exchange workshops. For example, also today's e-seminar, which David already mentioned, is brought to you in collaboration with the Pharma Connect Africa channel. Okay, and next, one last note before we start today. Please note also our annual conference is coming up actually in July in Poland, Krakow. And this will be a very special event this year because we have teamed up with many other societies like the Phytochemical Society of Europe, the French and the American Society of Pharmacognosy and others. And also for the African community, there will be a pre-congress event where we will focus on African herbal medicine. So David, for example, will give a keynote, but we would also like to get to know what's on your research agenda. So we would be very happy if you submit an abstract and participate and get into exchange with us there. You can do this either in person in Krakow, directly in Poland, or also tune in online. The links will be provided in the chat. Okay, but coming now back to our business of the day. As I said, we want to promote an evidence-based use of medicinal and natural products. So today we will illustrate how this could be achieved and also talk about the challenges that you and many others are facing. So for this, we have prepared two cases. We are starting with a phyto drug, which is indicated for the use in sickle cell disease. This was investigated and developed by the team around Professor Sylvain Ouedraogo, and he will present the development and the preclinical data set in the first talk. And afterwards, we will hear about how this data set can actually be used to design a clinical trial. This has been done 2 years ago, and Professor Sylvain Ouedraogo was supported by Merlin Wilcox, who is our second speaker, and will report about the developing clinical trial design. And then another case will in the end be presented by Professor Matsabizia, who will also use this moment to talk about opportunities and challenges of trials which are dealing with herbal medicinal products. Okay, so that's the idea for the day. And I would ask Sylvain to
Yes, I'm here.
Yes, activate his camera, and you can also start sharing your screen. Sylvain is the director of the Research Institute of Health Sciences in Burkina Faso, and he is specialized in pharmacology and ethnopharmacology, and will present us one case in the first talk. Sylvain, the floor is yours.
So thank you. Thank you, moderators. First, I would like to thank African Research Network of the Society for Medicinal Plants and Natural Product Research, and also thank Pharma Connect Africa for this meeting, for this seminar, bringing together experts in phytotherapy research, especially to exchange on the clinical test of phyto drug. These clinical tests are the last step of development of phyto drug for us with a number of challenges and with many, many difficulties, many. So I return to present you a fighter drug, FACA, that we use for the management of sickle cell disease. It's in a capsule form, dosed at 80 milligrams for children and 180 for adults. So here the background. Sickle cell disease is a genetic disease that affects hemoglobin and red blood cells, and it's a major public health in Burkina Faso, problem in Burkina Faso and in Africa. 5% of the world population carried a sickle cell gene, and this can reach 10 to 40% in some part of Africa. In Burkina Faso, this disease affects about 2% of new births, of which 50 to 75% die before 5 years old. And for the treatment, for the serious form, it's based on hydroxycarbamide, morphine and morphine, non-morphine analgesic, repeated blood transfusion, vascular filling, and here in Burkina, FACA as a phytomedicine. There is also some curative treatment, like hematopoietic cell transplantation. And there is also a preventive with information and education of couples. So here in Burkina, we developed a fighter drug called FACA. This recipe is from a traditional healer. And during a regal internship, that's come back to us that a healer was successfully treating sick children. And after clinical and biological follow-up of the patients, it was realized that there are sickle cells patients. And the tight collaboration with the healers allowed us to identify 2 plants, Xanthosylum xanthosyloides and Calotropis persera. The recipe is the mixture of these 2 plants. It's a powder from a root bark, and the traditional healer administered it orally. So after the observation with the healers, we realized ethno-medical evidence, and this time in the hospital. The study focused on 40 sickle cell children, divided into 4 groups, 1 for FACA, 1 for Calotropis persera, 1 for Xanthosylum xanthosyloides, and the last one for Idergine as a reference. And this is 1995. And a second study occurred in 2021 on 20 other subjects. But this one was in crisis with vaso-occlusive situation. And for prevention treatment, 1 capsule is administered daily. And for crisis, it's administered 3 capsules per day. And then we separate the 2 plants to better understand the potential benefit of the mixture. And the evaluation of criteria was as follows: pain in the patient complaint according to 4 degrees of pain, deadline for full crisis amendment, crisis interval, side effects, and sickle cell rates. For the curative treatment, the results are therefore, we observed a disappearance of asthenia and pain at the end of treatment. Biological signs of inflammation return to the normal value, and the sickle cell count dropped significantly. The rate of sickle cell disease decreased from the first hour until the end of FACA treatment. For the prophylactic treatment, you observe depletion of anemia in heterozygotes and disappearance in homozygotes after 1 month of treatment. The number of red blood cells, leukocytes, platelets, and hemoglobin level remained stable. No crisis was observed during preventive treatment with FACA. And we observed that FACA is more effective in any way than the isolated plane. Take it alone. So these results are therefore lead us to seek the potential mechanism of action according to the hypothesis based on the physiopathology of sickle cell disease. This includes cycling, pain and fever, vascular occlusion, inflammation, and blood viscosity. So first, we realized the anti-cycling study on in vitro cycling induced by sodium metasulfite, the ML test. And the test revealed an anti-cycling property of FACA and these components. We observed 80% reduction rate of sickle cell anemia after 5-minute contact with FACA. It is even more effective than idergine, administered as reference. And the anti-cycling activity of FACA is also dose-dependent, with a greater effect than the two isolated compounds, the two isolated component of FACA. And after that, a bio-guided extraction of xanthocylum xanthoxyloides revealed an anti-cycling activity led to isolated 3 isomers of vanillic acid. And these new molecules isolated from root bark of xanthoxylum xanthoxyloides, we call them brokinabine A, B, and C. And these molecules have anticycline activity. This property is growing from brokinabine A to brokinabine C. So the test of the anti-inflammatory property is carried out on the model of edema on mouse paw induced by caragene. And we see that FACA and its isolated component induce an inhibition of edema, and thus indicate an anti-inflammatory properties. This activity is also dose-dependent with a greater effect with FACA. An analgesic activity here is performed on 2 models. Acid acetic induced pain in mice, that's the peripheral pain, and hot plaque induced pain in the rat tail, the central pain. And the observation showed that only peripheral pain is inhibited by FACA and its component. And with regard to the inhibitory doses 50, FACA seem even more effective than Fagara xanthoxyloides and calotropis porcera. So FACA is a peripheral analgesic property and not central property. We also evaluate and Yeah, we also evaluate was that the antipyretic properties. The antipyretic properties is evaluated according to Saccharomyces-induced fever in mouse. And here we observe that FACA and its component induce inhibition of fever induced by Saccharomyces. And when looking at the inhibitory dose 50, FACA has a better antipyretic activity than its component. That reveals a synergic effect of FACA. We also revealed that FACA induces a vasodilator effect here, partly dependent on the presence of the endothelium and partly by direct action on vascular smooth muscle. And we clearly observe that the function of the endothelium is mediated by nitric oxide. And this information can be interesting in the case of vaso-occlusive crisis during sickle cell disease. So we also evaluate the safety of the product. And this assessment follows several models, including acute toxicity in rodents, in intraperitoneal and oral administration. For intraperitoneal administration, Fagara xanthoxyloides is virtually non-toxic. FACA and Caltrate procera is moderately toxic according to recommended classification. And the oral lethal dose 50 is greater than 5,000 milligrams per kilogram body weight. So we are looking at a relatively safe product, but it is often described the presence of cardenolite in the Calotropis procera, and this with a potential cardiotonic effect. But our observation in what do not indicate a change in blood pressure or heart rate, rather than to observe a positive inotropic effect on isolated heart. And more recently, in 2022, we evaluate the 90-day chronic toxicity of FACA and genotoxicity and reproductive toxicity. So in chronic administration of FACA is well tolerated at the doses higher than the dose of use, the dose of therapeutic use. It shows no mutagenic effect on erythrocytes. And on reproductive situation, no significant adverse effect, except here a decrease in fertility of female rat, unfortunately. But it's a reverse effect in the male rat with this, the dose-dependent stimulation of spermatogenesis in the male rat. So for the chemistry, it's noting that this product is rich in chemical compounds such as sterol and T-terpene, alkaloids, flavonoids, coumarin, and tannins. And with regard to Fagara xanthoxyloides, several molecules of interest are identified, including bukinabine, with their anticycling properties, I would say. So we have retained cardenolite from Calotropis procera and vanillic acid from Xanthoxylum xanthoxyloides for the quality control of the raw material. So we have protected FACA as a trademark, and FACA obtained, I think, several authorization of marketing authorization in Burkina Faso from 2010 to now, and last year in Côte d'Ivoire. We also realized a pharmacovigilance study in 1995 and also in 2023 on 132 patients, and the second one in 201 adults. And this reveals some side effects, and it's mainly about nausea, diarrhea, vomiting, and abdominal pain. But the efficacy ratio versus adverse effect is in favor for FACA. So FACA induced a crisis amendment in sickle cell patients, probably by anticycline, antipyretic, and analgesic effects. Its vasodilating property can confer a beneficial effect in the situation of vaso-occlusive crisis. So this is the sum of biologic effects that will be contribute to therapeutic activity of this product. And this research revealed a synergetic effect of the two plants that indicate the interest of the mixture, like the traditional healer used. So for the future, administration of capsule is difficult in infants from 0 to 5 years. So the capsule must be open and to put in the porridge or water, for example. So we are therefore proposing the syrup form, which is in clinical trial right now. And for the capsule form, we are also considering a full clinical trial. So FACA wins several prizes. And the last one is the first prize of Gallian Africa in last year for the best herbal medicine product. And we received this prize from the President Macky Sall of Dakar in Senegal. So I would like to kindly thank you again, the organization of this seminar. Thank you, GA. Thank you, Pharma Connect Africa. And thank you, moderator.
Thank you, Sylvain, for the excellent talk. I think you showed us very successful example, covering, I would say, all the aspects. Deep research, of course, herbal medicine first, chemical composition, safety, mechanism of action, including gender aspects also, pharmacovigilance. Thank you very much. And now it's time for 2 questions because we have to respect the time frame. So please, 2 questions to Sylvain. Otherwise, you can use, of course, the chat box for your questions.
Yeah, so there's a question here from Bernard Texon. Are there any contraindications in the use of FACA? How much is it in the marketplace?
Yes, in the market, the male contraindication is mainly in a woman, a pregnant woman, because we didn't have information about this situation. So there is a contraindication for this, for pregnant women only, for instance.
Okay, and then from Mikhail, is the effect of FACA symptomatic only? Is there a chance to improve the life of patients in the long term?
No, it's only for the management of sickle cells. And you have to take it, anytime when you have crisis.
Okay.
Yeah.
All right.
We
Maybe
also have a raised hand in the
a
audience.
raised hand, yes. From Frank Landry, you can do your question, please.
Thank you, madam. Thank you, sir, for your nice presentation. I really find it amazing, and I have one preoccupation. It's about the FACA. Does it made from plant? If yes, from which plants?
Yeah, it's from 2 plants, I say. It's from Xanthosylum xanthosyloides and Calotropis persera. It's a mixture of these 2 plants.
Okay.
I hope I understand your question.
Sir, how did you tried any other plants beside these 2 plants, and how did you came up with like there would be many different parts of the plant that could have been useful, right? So you just selected only. Did you went by what is that called? Bioactivity guided fractionation, or there is some other approach?
So we didn't evaluate any part of these 2 plants. We just developed like a traditional healer use it. So we didn't evaluate any part of this plant for this efficacy. But no, we use all the powder. It's not a fractionation. In the capsule is all the powder from root bark of these 2 plants.
Okay.
I hope I understand you.
Can we move on to the
Yes.
next speaker? You
Wait.
can put your message in the chat box.
Okay, now it's time to proceed, and I would like to ask from Dr. Maylene Wilcox to take the floor, the cyber floor, and give us his talk for Guide of Development of Clinical Trials and Design.